Today we have our last of our introduction type lessons, and hopefully the last time you have to listen to me talk for the majority of the class period. And so today we kind of get started in prepping for our first assignment. As far as our first, like big writing paper, there's 3 big writing papers, obviously the one real big one, 2 prep ones, and then there's a little reflection one. This first one that we're going to be doing is this critical appraisal. And when we talk about critical appraisal in the field of evidence-based practice, what we're really talking about now is basically how do we evaluate the research for applicability. It's going to be a lot more than just, good, bad, right, wrong. Because as with everything else, the answer is always, it depends, right? Is this a good research article? Well, it depends, right? That's just how reality works. So some of this may be review for some of you. Hopefully it's not brand new to anybody. And we will go through everything that we go through today. We will repeat on Wednesday, but specifically for our assignment. So think of today in this lesson as the general overview. Just sit back again, kind of try to gain 1 or 2 points. On Wednesday, we will then look to do this for a specific article. Between Wednesday and Friday, you will write up your critically appraised paper. And Friday, we will do peer review. Sounds good? All right, so let's start first off with this, how to read research. And basically, this is just an introduction to a research article. The nice thing with this is because we're reading structured research articles, we can expect the authors to have done a lot of the organization work for us. So we just need to find the pieces that we're looking for. When we look at reading and research, we're trying to pull out the general who, what, where, when, why, how sorts of questions. And so that's where we need to do. Look at where these things are. So, the abstract, nowadays even the abstracts are structured, which is nice. Gives us basically enough information to see whether or not we need to or we want to read the article. That's the purpose of the abstract. Right? Anything written in the abstract is so out of context that it's really not worth even reading if we already know that this article applies to what we want. Right? So when we think of the abstract, only think of the question, is this worth reading the article? That's it. After that, we're going to look at the specific headlines associated with the different topics. So here's basically a flow chart of how we can go about figuring out what we need to know from each article. So when we look at the title, right, titles are supposed to be descriptive and directional now. And what we're really just looking at here is, does it make sense? Is it possible? Does the title give us enough information to warrant reading the abstract? The abstract then gives us that snapshot. The introduction gives us the background. So the idea with the introduction, and the only reason to read an introduction, is if you're not convinced or you don't know enough about the problem to decide whether or not it is in fact a problem worth researching. Okay, if, for example, you're looking up and you're trying to investigate the effects of recovery in baseball pitching, you already know that there's a problem there. You've probably researched it a little bit on your own. The introduction is completely useless at this point. Right? If there was ever a section to skip, it's the introduction. Now that goes contrary to the way most people read research. Why? Because what do most people read the introduction for? So they don't have to read all the other articles, right, that talked about that. I can read, if I read the introduction in one article, then I don't have to read the other articles. There's only one problem with that. And the problem with that very much is you're relying on somebody else's interpretation. And that can get you in trouble. So in evidence-based practice and in exercise science writing, it is never acceptable to secondarily cite an article. So what that means is if I see a stat or something in an introduction or a discussion, and I would like to include that in my paper, it is only acceptable to do so as if I have gotten and read the other paper. Does that make sense? So if an article shows up in your reference page, the assumption is that you have read that article. Okay? Methods, right? Methods is usually the second to last section that people want to read. The worst section is the results, right? I mean, anybody ever try to read those results sections? You know, they're a pain. And I would agree. And we're going to skip a lot of the results because really what we're going to go for is the tables in the results. But the methods are by far the most important section of the paper. And the reason for that is the methods are going to answer the question of, is this study applicable to you? And it does that by describing most of the who, what, when, why, and how. So if I was reading a research study and I'm trying to think about whether or not this applies to me, probably 75% of my time would be reading the methods. About 15% of my time would be looking at the results tables. And then maybe 10% in the discussion and introduction, which of course is the exact opposite from what you usually do, right? I mean, how many people have ever cited something from a methods or results section of a paper? Right? Assuming you don't just look at the abstract, right, which we don't. I mean, that's not who we are, right? We read papers, we don't abstract hunt, right? Never cited by only looking at the abstract, right? Never. That's not who we are, that's not cool, right? But then usually, hey, what kind of, things can I nab from the discussion, right? And so the results answers the question that the research team created using all sorts of statistics and different things. Most of that information in there is very technical and has to do with the stats. You can skip most of that. There are two things that you really want to look for from the results section, right? And when you're going through and you're doing these for your papers here, these are the two things you want to find. First is the raw data as much as possible. So usually there's a table there that shows the data between the groups. You guys are all smart people. You can then look at that data and see, is this important? One thing to keep in mind with this is the term significant in research means statistical significance, which is simply the level of confidence that there is in fact a true difference between the groups, opposed to a difference based on chance. Significant does not mean important. If you test enough people, you will see significance in there, right, between the groups, but that doesn't mean it's important. The statistic that does talk about importance is effect size. So if you ever see effect size listed in the results, now that's telling you how big of a difference relative to clinical significance is there. And basically, effect size is the difference between the two groups over the standard deviation. And so when we're looking at this, if we're looking at something that has very small deviations, right? For example, let's talk about like winning 100-meter times in elite athletes, right? There's a very small difference between, you know, first place, second place, third place, all those sorts of things. Small little deviations are important. So therefore, something that improved time by, say, half a second would be enormous. Whereas if something improved range of motion, say, by a half a degree, completely irrelevant, right? Or using the same example, if you save a half a second on a mile time, not that worth it, right? So effect size will give you that main difference. And so when we're looking at reading a research article, the discussion then, as the last, is the completely useless area. And the reason that it's completely useless is because the discussion is the author telling us how their results fit into the greater world, right? Both their situation as well as the research. But we don't care about their situation, and we're not particularly interested in the research, right? We want to know whether or not this article will affect what I'm going to go do out at practice today, or what I'm trying to come up with, right? So the discussion is generally useless. When we're going through reading research, the most important thing is, do the methods match something that is relevant to us? Are the subjects similar? Because if the subjects are not similar, it doesn't really matter as much, right? Are the measures appropriate? All right. Are their outcomes something I care about? And then finally, in the results, is there real difference that's meaningful to me? And so that's the generic how to read a research article. The rest of this time is we're going to talk about how do we figure this out. So the first one is the hierarchy of evidence. Has anybody had a lecture on hierarchy of evidence? If I mention that term, does anybody know what I'm talking about? Okay, good. That makes life easier. So when we talk about hierarchy of evidence, just think of any other hierarchy you're familiar with. A hierarchy defines who is better or more important than others in a specific situation. So that is the most important thing to continue to keep in mind as we go through this hierarchy, is hierarchies are situationally dependent. So we talk about this in general because we're going to talk about methodological quality primarily. But there are situations where it might be inappropriate to get higher levels of evidence. We'll see that one of the highest levels of evidence is a randomized study. Well, it would be unethical for me to want to do an ankle study where I'm testing like the efficacy of ankle bracing, and I randomize determine whether or not I'm going to give you an ankle sprain. Right? If I got a whole bunch of healthy people and I go, okay, you get an ankle sprain, you don't, you do, you don't, you know, like that doesn't work. So obviously that would be inappropriate. So even though we would see a lower level of evidence in that situation, it's still specific to that situation where that might be the highest level of evidence possible. Okay. Following that, the hierarchy of evidence pertains to the methodological quality. But even more important than the methodological quality is going to be the relevance of the evidence to the problem. That's why we start out in the abstract by not reading for whether or not this is a high quality study, but we start by reading whether or not this study is relevant to us. So don't ever forget that as well. Okay, there are different types of level of grades for level of evidence to make life easier for us. I just picked one that's one of the most common. There are more, but I didn't want to confuse us with multiple things that are basically saying the same thing. So we're going to use the Oxford Center for Evidence-Based Medicine, or you'll see it abbreviated a lot, OCEB, or OCEBM. Okay, this is going to primarily differentiate between strengths of levels of evidence based on sampling and their research methods. So here is the levels of evidence. There are 5 levels, and each one has subcategories. 1 is the highest, A is the highest, okay? Makes sense. Well, I guess not really, because 5 is greater than 1, but 1 is always closest to 0. Right, whereas 5 may be the highest, but in another one, 6 might be the highest. So that's the idea. So let's work our way through this. The first one that you need to understand is in a lot of the subgroups, there's this idea of a systematic review. That's what we're going to be writing. What a systematic review is a research study of research studies. It has methods, it has results, but its subjects are different research articles. So if you've read reviews before, right? And a review is simply what somebody else says about the literature. The biggest question is in a review is what literature did they review? Was it comprehensive? Was it selective? Whatever. What a systematic review does is a systematic review eliminates that decision process. You set a methodology, and then every research article that matches that methodology is included in the review. Therefore, it's considered the highest level of evidence in that category because it includes multiple research articles, and you can't cherry pick them. The only exception in there that you might see is some systematic reviews also include a meta-analysis. You ever read a research article and see the term meta-analysis? All a meta-analysis is they then rerun the statistics and pool the results. All right, so systematic reviews, meta-analysis are going to be the highest. RCT is a randomized controlled trial. When we look at research, and as we go through in our critical appraisal, you'll see we'll spend a decent amount of time looking for what we call like confounding variables. This is anything else that might have caused a difference between the groups besides the intervention. The biggest way to account for confounding variables is through randomization. The idea that if I randomize who gets assigned to what condition, then most of those other variables will come out in the wash. We would have about an equal number of men and women. We'd have about an equal number of tall and short people, about the same number of flexible versus inflexible people. And we can double check that by looking at the results section before baseline. But randomized clinical trials is going to be a level 1 evidence because most of our accounting of confounding variables will be accounted for through randomization. We will talk about the Pedro scale later, but the Pedro scale is a way because the randomized controlled trial is the like gold standard for research. So that's what most of our research should be. So in order to differentiate between higher and lower quality randomized controlled trials, we have this Pedro scale that will do that for us, and we will actually spend an entire day on Pedro's scale. So level 2 now is cohort studies. And what cohort means is basically a pre-existing group. Now we can have prospective or retrospective studies, basically meaning, do we create the group beforehand or after? But the group exists in isolation of the study. So going back to my ankle sprain example, I can't randomize them to whether or not they sustain an ankle sprain or not. So I would have to do a cohort study of individuals that have sprained their ankles and individuals that haven't sprained their ankles. And then I try to account for everything else in there. But there could be underlying factors on why some individuals have sprained their ankles, why others haven't. All right, and that's just part of why that level of evidence is downgraded. All right, for example, if I went and looked at, people and I just grabbed people from Augustana Athletics that have sprained their ankles and those that haven't, my bet is I would get more basketball players in the sprain their ankle and more track athletes in have not. Right? Well, and then that would make sense then if I was then going through and looking and saying the average height, right? I might say, people that have sprained their ankle tend to be taller than people that don't. Maybe it's, being tall causes spraining ankles. Well, not really. It's just based on my sample. I got more basketball players, and basketball players tend to be taller than track athletes for obvious reasons. Does that make sense? How we're decreasing the level of evidence? But remember, going back, this is simply a decrease in the level of evidence, but it still needs to be considered within the applicability of our study. So for example, if I'm trying to look at the effectiveness of an ankle training program, right, to strengthen people's ankles, it might be better for me to look at a level 2 cohort study, right, that compared ankle or people with injured ankles and those that weren't during a program than somebody that only used healthy individuals but randomized them. Right? We first start off with applicability. Then with applicability being equal, we go to level of evidence. Make sense on how we're using this somewhat? Okay. Levels 3 and 4 kind of come intermixed in there. Basically what these are is these are non-experimental studies. They don't necessarily have a comparison group. Sometimes it's because we're doing things that warrant universal treatment. Sometimes it's because we're doing smaller groups. All right, and what each of those means are described there as well. Lastly, at the bottom of the level of evidence are bench research, animal studies, and expert opinions. Now, the reason for this is because when we get in this bench research or basic science, we're looking at things that are usually not directly applicable to our human population. So going back to the example I used before on ultrasound and increasing muscle temperature, right? That is bench basic science research. Because the goal is not to increase muscle temperature, the goal is to increase range of motion. Right? And so we're looking at a sub-goal of that. Animal studies, for obvious reasons with that, is the good thing with animal studies is you usually can randomly assign animals to injuries. The problem is you're using an animal model. And anytime you use a model, you're only as good as the assumptions built into the model. And the basic assumption is that the animal's physiology is similar to humans and that they're going to heal in a similar way. And then expert research, expert opinion is just that, right? It's expert opinion. So this chart here is worth taking a look at in your own time as you want if you have specific questions. But this also goes through and talks about how different levels of evidence are appropriate for different types of conditions. So this goes one step farther and takes it where it's like, okay, in a study, right? Let's look at like a biological sex-based study. There's no such thing as a randomization of biological sex, right? Not possible, right? So talking about a theoretical level 1 that doesn't exist is a waste of time. So that would then just switch down to like cohort studies. Okay. Then from that, right, that's how you evaluate each article. So each piece of research has its own level of evidence, right? So when we go through and we do our critical appraisal on Wednesday, we will assign that research article a level of evidence. However, we're rarely going to be making decisions based off of a single piece of research. We're going to be combining them. And that will be not this week's assignment, but next week's assignment will be synthesizing research. When we start synthesizing research and we start combining more than one article, we then have what we call, grades of recommendation. And to not confuse us, these are A, B, C, and D. Follows pretty simply. A is consistent level 1 evidence. B is consistent level 2 evidence or inconsistent level 1 evidence, but trending in a specific way. C is level 4 evidence or inconsistent level 2 and 3. And then D is level 5, inconclusive or absent. Okay. We will look at this more when we get there. All right. I said that I wasn't going to talk about this other ones, and so that was sort. It somehow stayed in there. Okay, so application of critical appraisal. Okay, when we get now into this, now we need to apply it. So we've talked about it in theory. Now we're going to apply it and how we do that. And how you apply critical appraisal is you use it in the evidence-based practice format. That's all we're talking about. But as I promised, the evidence-based practice format is a way of doing things, not an outcome. So there are tools here to help you get that. So when we go through and we're trying to determine what level of evidence this is, as well as its clinical applicability, we're going to be answering these 3 questions. Is the study valid? Is the study reliable? And are the findings of the study clinically relevant? 2 of these have to do with the methods. 1 has to do with the results. Kind of hence, my 75-10 mentality. Or I think it was 75-15-10, right? Mainly, we need to look at the methods. We will then also get into the results. But we need to know what these terms mean and how we answer these questions. So when we talk about, has anybody done internal, external validity, reliability discussions, or is this brand new? A lot of brand new. All right, so in this, there's about 100,000 resources on, validity and reliability things. One thing to keep in mind is if you're looking up this and trying to get more examples, these terms are similar or are the same and are used throughout scientific research, but their applicability and how they get used differs slightly. So make sure you're looking more in the exercise science, sports medicine realm with that. But it's relatively simple if you just think big picture. So internal validity, right? If I think of internally valid with myself, right? With internal validity, I want to know how true to myself am I? So when we look at internal validity of a study, we're looking at, does the study do what it says it's supposed to do, right? Does it answer the question it's supposed to answer? And you can look at that throughout each different area. So when we look at the different things, I'm gonna, All right, come back to here. When we look at the different areas for is a study internally valid, we're going to look at all of those, what's and how's within that and say, are those in fact valid? Okay. Did they in fact measure what they said they were going to measure? If it says I'm measuring power, right, which often comes into play, it says I'm measuring power, right? And then in that measurement, they do a 3-rep max. And make the argument that that's probably not the best measure of power, right? Something like a vertical jump would be a better measure of power. But what you'll see is they will continue to use the word power, 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 but what they mean is 3-rep max. So that'd be an inconsistency in the internal validity. Does that make sense? There can also be confounding variables. Any of those confounding variables that we thought of, is there anything else within the research that could create a problem so that way it's not just the independent variable affecting change? Some of the common with that is selection bias, right? Like, is there some way that the groups got messed with somehow? Even if it's not on purpose. Like if I was doing that study, that same study beforehand on the ankle sprains, and I did random selection, but for whatever reason, I got a bunch of tall people in one group and a bunch of short people in another group. Now I have 2 variables that could affect it, right? I have my intervention and I have height. Height would be that confounding variable. Right? On that same note, it is quite possible that the group that they selected is inappropriate for their reference. And this is where healthy versus involved participants or injured participants are. If I'm trying to increase range of motion, it would be inappropriate for me to study people that have full range of motion. Right? Because it would make sense that it would be difficult to increase range of motion in people that already have full range of motion. But that happens all the time, right? They use healthy people to evaluate the effectiveness of a treatment based for people with specific conditions. That's a knock on the internal validity, right? Delimitations are basically decisions that the investigators make that affect that. So, there can be, inadvertent things, but then there are just delimitations, like studying healthy people. That's a delimitation. I made the decision to study healthy people, but that's going to limit the effectiveness of my intervention to be true to what it's supposed to be. Blinding, we'll see blinding a lot when we talk about Pedro's scale. Blinding is simply the idea that the subject and the researcher, or some combination of that, as well as the evaluator, don't know what condition people are in. Right? And for obvious reasons, there can be anything from overt to subliminal changes associated with knowing whether you're in the group, right? The whole placebo effect sort of thing. External validity then goes for whether or not if I use the example of me, whether or not I am representative of other people. Right? So internal validity is whether or not I am true to myself. External validity is, does looking at me have anything to do with looking at other people? And the main thing with this is going to be the applicability of their subjects to other subjects. So oftentimes when we look at when we look at that, it's going to be, if we only studied college-age kids, does this really have anything to do with elderly populations? Right? Probably not. That's external validity. It says exactly what will happen with college kids. That's its internal validity. It's true to its question, but it may not be applicable to other things. Validity of measures, this is more internal validity type stuff, right? This is all sorts of different ways that we can establish validity, but basically this says is, do the measures do what they say they're going to do, similar to internal validity. Okay. So how we can go about appraising the validity of a study, it's going to depend on the design. And these are the sorts of questions we will work through on Wednesday. Reliability is just purely reproducibility. Are the methods that they used reproducible, right? Are they specific enough in their ability to measure differences? And are they used in a way that is appropriate? So there's air, right? Nothing measures anything exactly, right? You know, that's why, you know, like you pick scales, right, that tell you what you want, because they're not all perfectly calibrated. There's amount of air in there. So if you want to say you lost 5 pounds, just keep going around the scales at the gym till you find one that measures you a little lighter, right? That's just the amount of air in there. Even at that same note, if you were to get precise enough, each time you would get on the scale, it's not going to be exactly the same. There's a certain amount of air within that. Right? Reliability things, usually you can count on the reviewers to identify any reliability issues. If a study isn't reliable, it probably isn't published. Right? And the flow chart for how we will do that. Applicability, right? When we get into applicability, we are now going through and just answering the question that we started off with at the abstract. Does this apply to us? Are the results different enough to warrant me using this intervention? Right? That's what we're looking at with applicability, as well as a little bit in that external validity section. As far as resources, all these sorts of different things are what we will look at on Wednesday as well. So we are going to do a critically appraised paper, which is one article. You can also do a critically appraised topic, which usually is about 3 to 5 articles. So what I would like you to do is, for Wednesday, there is a chapter that I posted from a book on Canvas that will explain how to do a critically appraised paper. It will answer these questions. These are what a critically appraised paper is, just this, answering a paper written answering these prompts. Okay, take a look at that. As well, we will all do the same research article that I will give you on Wednesday. For this one, you do not find your own. For this week and next week, we're all going to be working together, so that way we can focus on the process, opposed to trying to figure out how you applied the process to your research. So this week and next week, we'll all be doing the same stuff. Other questions? Very good. That being the case, have a great day, and I will see you Wednesday.